Well, ladies and gentlemen, welcome. This is the first webinar that is sponsored by Connecticut Valley section of the American Chemical Society. My name is Shurab Chakraborty. I am the current chair of the local section of the ACS, and I will moderate the session. If you have any questions during the presentation, please send it via chat. We'll try to compile them and then present them to our speaker. At the end of those questions, you can raise your hand and ask questions, unmute yourself at that point. So that's how we're going to run the presentation itself. If you have any questions about the local section or our yearly activities, please be in touch with me. I'm going to send our website address via chat. You can save it and you can mark it so that you can get back to us. We are always enthusiastic, looking for enthusiastic volunteers, and we'll deeply appreciate your time commitment. If you feel like you are especially interested in any particular event, please reach out to me. My email address is in the website also. The executive board will be very, very happy to hear back from you. So that's the announcement part. Now to business. Please join me in welcoming today's speaker, Dr. Vaneet Sharma of International AIDS Vaccine Initiative. I know Vaneet for about 14 years. He graduated from Guru Nanak Dev University in India and pursued graduate studies first at University of New Orleans. That's where I met him. His tenure there was quite short. We all get displaced due to Hurricane Katrina, and then Vaneet moved on. He came to UConn, take a master's degree in chemistry, and then moved on to Northeastern University to take a Ph.D. Immediately after completing his PhD, he started working for Novartis Pharmaceuticals. There he worked on vaccine. And then after 3 years of tenure, he started working at International AIDS Vaccine Initiative. He has been working on vaccine for more than 8 years now. By training, I know that he's an analytical chemist, but all his work is in the interdisciplinary scientific areas. Today, he will talk and educate us about vaccine development. Vaneet, the floor is yours. Please take it away.
Okay, so welcome everyone. Am I audible enough? My presentation is visible?
Sounds great.
Okay, so let me go to this slide, this one, and then, okay, everybody is able to see in the big board? Okay, so thank you very much for giving me this opportunity to come and present and talk on this webinar, where I can discuss some of the work which we did do at International AIDS Vaccine Initiative, but then more about giving a generic overview of the development, cGMP manufacturing, and regulatory consideration. I modified my title a bit. I make it more specific. I'm going to be discussing more recombinant protein vaccines. And thank you, Dr. Shurab Chakraborty, for giving me this opportunity to come and talk about this. As an outline, I'm going to briefly touch upon the history of vaccines. Vaccines have a very rich history, so we're just going to give a brief overview. Different type of vaccines. The way I put this slide together and a few other slides, which I normally wouldn't have done, is because we are living in a very strange time, COVID pandemic, and I decided to give the different type of vaccines, trying to explain different type of vaccines based on the vaccines which are being developed for COVID. And because COVID has been such a big part of our life for last year, and it's an ongoing pandemic, so I thought that would be the best way I could discuss various type of vaccines. And over here, because COVID is a viral, disease, a viral, virus. So I'm going to discuss about different type of viral vaccines and which approaches, are being developed for it. Finally, the meat of my presentation is on the recombinant protein vaccines development and manufacturing. Now, I'm not gonna, discuss about research and how you design a molecule. That is not what I do. I am in a technical development manufacturing unit. So I'm gonna give you an operational viewpoint. And over there, I'm gonna impress you by trying to, show you that why it takes so much time to develop and manufacture vaccines. And over there, I will try to show you that what it makes to make a vaccine. And vaccine, as we all know, is one of those, if you could say, the molecules, the biological molecules you develop, which is for healthy individuals. And that is what actually makes vaccine a different ball together, a game ball, all different ball game together, as compared to any other therapies. That over here we have a protective medicine given to healthy individuals. And I will try to impress you why it takes such a long time, given the fact that we have already seen a lot of success in the COVID vaccines, which is on accelerated mode, but it typically takes 4 to 5 years of vaccine development and manufacturing. Again, we'll try to showcase some of the operational aspects of vaccine development and manufacturing. Then I will go and try to discuss the phase appropriate analytics, not because I'm an analytical chemist by training, which is yes, I am, but the reason I'm going to discuss it is that, analytics is one of the most heavy hitting, things in the vaccine manufacturing. They say that vaccine manufacturing is a process and product development. And you can only check the process and the product through analytics. So I'm going to go through, what sort of analytics is developed, what sort of assays are there, how you develop them, and probably trying to, give an overview of the analytics, the entire analytics you do for the entire campaign as the molecule enters development and it is released as a drug product. Regulatory compliance, I'm not going to go too much into it. Regulatory over here means FDA. Interaction with FDA always means a lot of paperwork. I'm just going to give a flavor of it, trying to impress what it needs to be done, or what the COVID vaccine developers must have done in order to make sure that the vaccines were developed and manufactured and released, and now, as you folks were discussing, already being given to individuals within a year. And it all depends upon the FDA, discussion with FDA, interaction with FDA, and how it is possible, how it was possible that something which is being developed over 5-6 years historically, how everything was achieved. It's a modern miracle. In terms of history, I think probably everybody of us must have read it, heard it, somewhere that in 1796, Edward Jenner, he developed the first smallpox vaccine, from a cowpox. It's again a first modern sort of vaccines, and the word vaccine has its origin from vacca, which means cow, and that is how the field started. It was a momentous work done by Louis Pasteur in 1800s, which really set the ball for the vaccine development, future vaccine development. And as you could see, many diseases were eradicated in 1900s, 1940, 50. Polio, BCG, diphtheria. All those are more or less diseases which have been overcome by human mankind. And then as we progress, last year, 2019 was another important year where vaccines for Ebola were actually authorized or approved by FDA. A vaccine by Merck was approved, which was the first vaccine for Ebola. And then just recently, a vaccine for Ebola from Johnson & Johnson, that was approved by FDA. And the reason Ebola became such an important, landmark is it is the success of vaccines like Ebola and success of the vaccines like flu vaccines, recombinant proteins flu vaccines, that many of these vaccines which are being developed by COVID vaccines, they have some sort of their background which relate to the work which was earlier done, especially very recently for Ebola and for recombinant protein vaccines for influenza. And now over here, this figure, I took it from 2018. Over here, there are different type of vaccine types. Now, what is a vaccine? Vaccine is a molecule, which is a part of a target pathogen. In this case, let's say part of coronavirus that is added to the human body, given to the human body in order for immune response to generate an immune response. And the basic science for a vaccine remains same, only the method of exposure varies. So typically, as you could see in this figure, there are different type of vaccines approaches. You can have a polysaccharide vaccine, which is a meningococcal vaccines are based on it from Pfizer. Whole pathogen, like where it could be inactivated virus, live attenuated vaccine. All the vaccines which we actually got as a kid, and many of the vaccines, which our kids get very early, when they are young, are the inactivated vaccines. These are the split subunit protein vaccines, recombinant protein vaccines. More recently, many of the flu vaccines are the recombinant protein vaccines. And over here in 2018, it was listed potential future concepts. And over here we have a nucleic acid based vaccines and vectors, structural vaccinology. And it's just a miracle that 2018 people were still designing and developing. And within 2 years, we have vaccines which are approved now for COVID. And these future concepts are no more future concepts, but a reality of the day. As I said in my introduction, that I'm trying to explain the different type of vaccines based on the COVID vaccine approach, because every COVID vaccines are so much in news, so much is being read, heard, and a lot of knowledge is already there. And most of as you were just discussing, probably some of you must have taken either Pfizer or Moderna vaccine. So I wanted to discuss that what sort of vaccines on this figure represent the COVID vaccines. So the whole pathogen, there are two vaccines not approved in US, but approved in India, China, and many other developing countries. One of them is Covaxin. It's a Bharat Biotech inactivated virus vaccine. Sinopharm, it's from China. And again, inactivated virus vaccine. Inactivated virus vaccine is a real virus, real coronavirus, which was rescued. And once the virus is rescued, it is modified either by chemical modification or its genetic makeup is modified and then formulated, by once it is inactivated, formulated to make sure that, it is no more capable to give the disease, but again, it's just potent enough to trigger an immune response. Again, most of the vaccines we got as a kid, many of the vaccines our young kids over in the US gets, it's the inactivated based vaccines. Relatively safe, effective, but again, till now they have not been approved in US. In future concepts, as I said, mRNA vaccine has been such a big news. There are two vaccines, Moderna, which is based in Massachusetts, and Pfizer-BioNTech. Pfizer is the manufacturing partner of BioNTech, which is a German company, and they came up with the mRNA vaccine, a very first-in-class type of vaccine. There have never been an mRNA vaccine in past, and this truly opened the floodgates for more vaccines for many other diseases, including HIV, for which we at IAVI are very excited about. Among the viral vector, there is a vaccine, Johnson & Johnson vaccine, which was approved just a couple of days back. It's a viral vector vaccine. It's a vaccine which uses a common cold, 8026 vector. And basically the goal is that the genetic information in the double-stranded DNA as a gene is smuggled within the big common cold virus, again, to make sure that it's not capable enough to cause the disease or any response other than the immunity we're looking for from the COVID virus. And over here, we have the Novavax, which is a protein-based vaccine. It's a split and subunit vaccine. It's a recombinant protein-based vaccine. It is being developed by Novavax in Maryland. It's still a candidate. They just finished the phase 3 trial in UK, got almost 90% effective, efficacy. In the US, the trial is ongoing. They're trying to get an emergency authorization, probably, based on the UK data. FDA may or may not agree. They might have full data for phase 3 sometime in end of May probably a very, exciting vaccine, generally very safe vaccines, protein-based vaccines which we have taken in past for many of the flu candidate, flu vaccine shots. So probably a vaccine which everybody, is waiting for, or like is a vaccine which will be available soon. Again, like I think the entire uh, the type of vaccines, especially the viral vector vaccine, they go back to the central dogma of biology, that the goal over here is to generate this protein, and I will tell you what sort of protein is this in human body. And the way it is given is, the way this is done is, again, the classical central dogma. Either you give the vaccine as a DNA, double strand DNA, it instructs mRNA to make a protein, or you actually give the mRNA, which Moderna and BioNTech is doing, to generate the same protein, or Novavax, which is actually giving the protein itself, the purified protein, so that immunity is, immune response is generated against those protein, and immunity is generated or developed on the exposure. On the mRNA vaccines, mRNA instructs our cells to produce the SARS-CoV-2 spike protein. Now these red guys, the one over here, these are the spike ones. And as I said, every approach over here in US, they are trying to make these spike protein in human body. And it is by making these spike protein in the human body, they are trying to generate an immune response. And mRNA, it directly instructs the cells to make the protein. Once the protein is formed, the body defense system, it recognizes the spike protein as a foreign and begins to protect against it, thus triggering an immune response, which is we all calling it an immunity. It's a simple formulation concept. The mRNA is synthetically generated, and every mRNA will have a 5' cap, which is a very important piece of it, 3' poly A, and then this green zone, this is the coding sequence. Now, the beauty of mRNA vaccines is that it is a synthetic molecule, very easily generated, and then once the coding sequence, probably all you have to do is that you have to change this green portion, and your mRNA construct is ready for the vaccine we are trying to develop against any pathogen. But these mRNA in itself, they are very sensitive to environment. So you just can't like give it directly to in the body. Over for this one, to help this problem, they actually formulate it with a liposome. Now over here, this is a petamic liposome, which is made up of cholesterol, lipids, phospholipids. And the entire goal is basically to encapsulate the RNA in this liposome. And that is done, not only it works as a delivery vehicle, but the goal is to make sure that mRNA survives in the body and is able to generate the protein. For two vaccines which are available, Moderna and Pfizer, the active ingredient is exactly the same ingredient. It is the same synthetic mRNA, because we're trying to produce the same glycoprotein. So active ingredient is exactly the same. There are very minor differences, but more or less it's exactly the same. It is the lipid nanoparticle, how the liposome is formed in the middle portion over here, which is different. And as I said, use of the cholesterol, the phospholipids, and various other things. We're trying to make that device, that carrier. And it is over here that a lot of exciting patent battles are going to be fought in future. The field of lipid nanoparticles, these devices, they have been generated, or they came throughout, from the research of various universities. A lot of work was done in Vancouver, then in Massachusetts, and over here there are some very exciting, patent battles which will be fought in future. So right now, because of the COVID, they have an agreement. They're not discussing much about it, but these are the things which are really who owns the patent for the lipid nanoparticle is going to make the money out of this COVID vaccine. Because in mRNA-based vaccine, it is that delivery vehicle, that liposome, which is actually the most important, because the mRNA is just a synthetic portion. And then, any vaccine has excipient. Over here, Moderna and Pfizer both have excipients, and the goal of the excipient is to make a right buffer system to ensure that the mRNA in the liposome is stable enough. When you stored it, the Pfizer BioNTech is stored at -80, Moderna is stored at -20. So you add cryoprotectant. You have to ensure that by adding all the salts and different other excipients to make sure that the vaccine is stable enough for long-term storage. Again, it is said, I heard someone said that he got the second injection. So 8 weeks, it is said after 8 weeks, the antibodies appear. So it is only by second injection that, the required amount of protein is produced in the body against which the trigger, the response triggered, and then the antibodies are seen. How long does immunity last? That's a question which everybody is trying to figure it out. So definitely for 5 to 7 months, because for the phase 3 trial, folks, they have seen that they have antibodies even after 5 to 7 months. Pfizer is claiming 2 years, but again, something which is very interesting to watch as more and more results come for the participants who took this phase 3 shot. Now, this is the single shot coronavirus vaccine from Johnson and Johnson, which was approved just a couple of days back. It is a AD26, a vector, a vaccine vector. Now, AD26 is a common cold, replication incompetent adenovirus. The whole goal is, as I told, that the coronavirus genetic information in genes through double-strand DNA is basically smuggled. So you took this AD26 virus and you change a portion of it and actually include the double strand which correspond to coronavirus in that bigger viral plasmid, and spike it or give it to individual, and then again the goal is same, that you have to generate that same protein, spike protein. Over here, this is actually, if you go to central dogma, this is you are giving basically a double strand DNA, not as such. There's another company based in Maryland, Innovia, which is actually giving a double strand DNA as such to generate the spike protein. But this is again the same exact, the double strand DNA in genes is given, and then in the body it instructs the mRNA to make the same spike protein. Coming on Novavax, Novavax is a purified form protein which is being given, Novavax NVX-CoV2373. It's a Novavax molecule again from Maryland. It's the glycoprotein as such, which is expressed in SF9 insect cell, baculovirus expression vector, and it is in form of a multimeric nanoparticle. It is not one protein, but it is a nanoparticle around 70 nanometer, and monomerically it's 180 nanometer, 180 kilodalton, and I think then it's a polymeric state is it's a higher molecular weight species. When they give this, each injection of this Novavax is actually going to be the purified multimeric nanoparticle, which is the glycoprotein, along with an adjuvant. Now, adjuvant is a very important facet for the protein-based vaccine. And there is a slide where I discussed the role of adjuvant. But over here, they are using their own metrics and adjuvant. It's adjuvant briefly means to aid, to help, to enhance. Basically, the protein-based vaccines are a very weakened form of vaccines. Inactivated viruses or viral vector vaccines, or even mRNA, they generate a higher immune response against them. Protein-based vaccines, because of the nature, of their nature, they actually have a weakened response if given by themselves. To increase their response, they are given backside mix with an adjuvant. In this case, an adjuvant is extracted from a saponin, from the saponate, extracted from the Puligia cephalora bark. It's a fascinating tree. If you folks are interested, I would request you to go Google it. It's a short form is QS. These are the trees which are found in Chile, and 1 gram of this bark or this this product is almost $200,000. So if anybody wanted to really do plantation, like they should really look for it. For some odd reason, only present in Chile. All the trees have already been bought by a company called Desert King in California, and this is a very important component for many adjuvants. Many of the GSK vaccines use this QS bar. Many of the Iskometrics Australian company, they use this bar, and Novavax is going to use many of the future vaccines. Actually, this is a miracle adjuvant. Everybody is looking for it. Probably the reason, the cost is $200,000 per gram for this bar. And as I said, just to briefly sum it up, in mRNA, you give basically in mRNA vaccines, you give and as well as in AD26, you give a code to the body to produce the spike protein. In the protein-based vaccines, such as Novavax, you actually are giving the purified spike glycoprotein. And now which takes me to the meat of my presentation, which is protein subunit vaccines, which are also called recombinant protein vaccines. And as I just explained, that rather than injecting a viral vector or a live attenuated, you basically are injecting with a protein subunit, the purified protein subunit, and they are nothing but the purified form of that glycoprotein. And generically, these type of proteins, they are considered very safe, as compared to live attenuated or inactivated killed vaccines. mRNAs are as safe as these vaccines, just because of the nature of the work. But then, as compared to the live attenuated or inactivated killed vaccines, these vaccines are comparatively, very safe. On the right hand side, I have 3 pictures. The Novavax, we just discussed about it. But then there are 2 pictures over here: BG505 SOSIP 664 GP140. So we produce this, we manufactured this. This is a molecule which came to us from Wheel Corner Medical School, Professor John Moore's molecule. And we did a phase 1 trial, got an exciting result. We published that work, manufacturing work over there. It's a GP140 trimer molecule, first of trimers ever produced and manufactured on the GMP scale. And now we are working on the second stage of this, the GT1 molecules, then the third and the fourth. So simultaneously, a lot of work is being done on these BT505 molecules, and a lot of trials, which we are part of, is being done for this molecule. Now, the middle molecule is the EOD GT860 This molecule was co-developed by IAVI and Professor Bill Sheaf from Scripps. And again, if you will see, this is actually very similar molecule to Novavax. So we developed this molecule. It's a 16-mer subunit, 2 million Dalton molecular weight, and it just finished its phase 1 trial. We got some really good results, which are going to be published soon, by Dr. Vilshi in one of the forthcoming papers. Interestingly, the recombinant protein vaccine, the world first recombinant protein-based vaccine, was actually approved by FDA in 2013. A company called Protein Science Corporation, which was in Connecticut, right next to where you folks work. And Protein Science Corporation now is Sanofi, and Sanofi has actually moved on from their egg-based vaccines to this recombinant protein-based vaccine. So a lot of the flu vaccines, like Sanofi vaccines, or even the vaccines which are manufactured in North Carolina by the Australian company, I forgot the name. They are the recombinant protein-based vaccines. Over here in this slide, I want to impress you that the question which everybody asks: why it takes so much time to develop and manufacture vaccines? And as I told you, that vaccines are one of those molecules which are intended for healthy individuals, which are intended for young kids, which are intended for newborn. The level of safety you have to showcase in vaccine is multifold higher than any other therapeutic molecules. Vaccines, as definition, are for protective immunity for healthy individuals. FDA is actually very, very strict on a lot of compliance issues. precisely the reason there are not many vaccine companies in the world. There are only 5 or 6 vaccine companies, and vaccine is a very cheap and cheap business. A lot of the vaccine companies actually they don't make much money, as compared to many other entities, because a lot of these vaccines are bought by government. But more importantly, like it's a very controlled field with a lot of regulation. And over here, I'm going to tell you about recombinant protein vaccine, and every type of vaccine have its own type of manufacturing issue. This slide is only for recombinant protein-based vaccine. mRNA is formulated, is manufactured in a very different way. Viral vectors are formulated and manufactured in a different way, but may or may not be similar to what I'm going to showcase over here. This slide again is only for the recombinant protein-based vaccine. So once the molecule has been designed in a university lab or in a research for research, and the molecule comes for the technical development, where you basically have to see that how stable the molecule is. It goes, the very first thing it goes through is the cell line development. In cell line development, everything starts with the plasmid. Plasmid is nothing but the DNA. So you get the DNA constructed, from a commercial entity, and then you transfect the DNA to the host cells. In the case of Novavax, the host cell is insect, based cells, but most of the work is actually being done on the million cell lines, CHO-based cells. Once the transduction is done, you get the pools, and from pools you get the clones, and in clones you have to actually showcase the single cell clonality. This is an absolutely must-have for FDA. FDA wants to make sure that the vaccine you have made, the protein vaccine you have made, the final product which came over, it came from a single cell clone. The way you do it is by doing limiting dilution, and over there you have to do it multiple times to make sure you take the pictures and you preserve those pictures. You have to, give those pictures as part of your package to FDA. And this is a single one reason where FDA can always come back and question you. They want to make absolutely sure that the final purified protein is from the single cell, so that there are no variants or there are no manufacturing issues when you go and do another GMP campaign. Once you select the clone, you do a clone development and characterization. In characterization, you basically are making sure that they are stable enough, and you do the testing to make sure that no adventitious agents are there, and that is part of the master cell banking. And once you have the master cell bank, that is actually preserved, because all the GMP campaign, and for many of the vaccine, it is not one GMP campaign. If there is a vaccine like a flu vaccine, recombinant flu vaccine, which are being generated every year, flu vaccine is not the right example, because the cocktail changes. On flu vaccine, sometime in August, 5 or 6 individuals, they sit in NIH, FDA, they decide which 5 or 6 strain, 3 or 4 strains they have to formulate. So probably not the right example, but then the master cell bank is important, because, even if the strain, one of the strain is being repeated every year, that strain has to come from this master cell bank, so that the company don't invest all the effort in doing this exercise all over again, and the entire thing can be manufactured in a very small time, as the case with flu vaccine. In flu vaccine, companies come to know around July, August, that this is the vaccine they have to come together for the flu this season, and they just have 3 or 4 months to manufacture that a vaccine. And probably everything comes back to that master cell bank, and I will tell you in next slide how much time it takes to make the master cell bank. A lot of these activities, which are known as process development, a process development has 2 pieces, upstream and downstream. They start before the master cell bank. Much before you have a master cell bank activity, you have a research cell bank, which is far enough to do all these activities. And over here for upstream and downstream, the goal is that, in upstream, the goal is to increase the titer, how much more you can produce. And in the downstream, the goal is to purify. So it's a very, tightrope exercise, where you want to increase the yield in upstream by changing the medias, by doing a lot of experiment. And simultaneously, more you are, producing, there are more impurities in it, and that need to be cleaned. And it's given in FDA regulations that all the vaccines, the recombinant protein vaccines, they have to go through a 3-step purification. After a harvest is available, from upstream, you clarify that harvest, and you go for first chromatographic purification step, which is affinity chromatography. That is followed by virus inactivation. Now, FDA wants that your purification scheme should be able to remove the model viruses. It is, even though the vaccine we get, it does not have any viruses as such, but there is always a risk because you are developing a vaccine in a cell-based system. There's always a risk there could be some host viruses, which could come along with the purified protein. Everything is tested, but just to be on the safer side, FDA want us to do the virus inactivation, followed by polishing step 1, which is ion exchange chromatography, normally either anionic or cationic depending on the molecule, followed by polishing step 2. Polishing step 2 is a nanoparticle, it's a 20 nanometer column through which you final purification is done. And again, the polishing step 2 is also the virus removal step. Focus is on safety to make sure there are no viruses available. But again, these are the demo steps, the things we have to do as part of regulation to ensure that even if there is viruses, which may or may not be there because of the system, they get removed. And once you do the upstream and the downstream development, you have a process log. Simultaneously to the process development activities, you are doing an analytical development. Now, in analytical development, there are assays which FDA want you to test your purified material. They want to have an appearance test, pH test. They want to make sure that their positive identity is there, the molecule have a high potency, the content, how much it is, how pure it is, do it have residual impurities. Then there are other safety assays. Once develop each and every assay, and these assays have to be based on the attributes of the molecule. They take some of these assays, take a lot of time, especially the HPLC-based assays. But eventually, all said and done, when it is done, you have an analytical log. Finally, when all these activities are done, you have a master cell bank, you have a process log, you have an analytical log. Then you go and make your demo batches. Your demo batches is the scale-up runs, in 50-liter scale or 25-liter scale. Then you do, once you test them, you find them, your purified protein is fine enough, you go and demonstrate consistency. Consistency has to be demonstrated by running the whole process 3 times at a high scale up, 100-liter run, let's say. But 100-liter run, 3 batches of 100-liter run has to be run exactly the same way, and then the purified protein is tested, and it has to be within the specification for it to pass. Once consistency is shown, you go and make a non-GMP engineering run. Now, this is at scale run. It is the same scale for which your GMP manufacturing has been done. It's a non-GMP run. And from that non-GMP run, you get your reference standard. The reference standard is the standard you're going to test your GMP against. Over here, you characterize it, you test it. You want to make sure that, the assays you are going to use for drug product cGMP manufactured material, the assays are fine. And then, you put this reference standard on a stability tested, intended, accelerated. The goal is that this material is actually the material against which the drug product is tested. So finally, everything is done. Then we go cGMP manufacturing. cGMP manufacturing is again taking a master cell, a vial from master cell bank, developing it, harvesting upstream, downstream, testing it, then finally releasing it. So these many operations are done, and every operation has complex controls. There are specifications that need to be achieved, and finally, all said and done, we have a drug product. How much time it takes? Over in this slide, I'm actually showing you very, very accelerated timelines. 4 years, 3 years back, 2 years back, the entire thing I'm showing you would have taken 2 to 3 years. Not because now we are cutting corners, but again, field is learning from work done previously. they are constantly in talk with the FDA. They go back and they tell them, should we do these studies? Can this be, can we leverage from the earlier studies and things like that. So over here, as you could see, cell line development and MCV testing takes solid 5 to 6 months. Process development is done simultaneously, again, solid 4 to 5 months. Analytical development, formulation development to make sure the assays are stability indicating. You do a lot of post degradation studies. You do small scale stability, a small, like intended stability study. The entire thing, even if everything is on a very fast pace, everything is working for you, this is like 6 to 7 months. I'm not even showing over here the consistency batches, demo batches, just because this is accelerated. If this would have been a normal program, you would have run a small scale, accelerated the consistency runs. But let's say everything went well, as they went well for the Novavax. They immediately went to the engineering and material at par. Engineering and material, they probably, if they would have done the talk studies, it would have taken 3 to 4 months. Probably they went back to FDA. They said that, they must have said that our platform has been used previously to manufacture glycoprotein. If you go and do the talk study, they're going to lose like 3 to 4 months. Should we or should we not? Again, it depends on FDA, what decision it took. But the engineering run in itself, it's a 3 to 4 months of campaign, followed by finally the cGMP manufacturing, which is a two-phase. Once you get the bulk, once you have a bulk, you actually aseptically fill and finish and do a primary and secondary packaging, and finally after doing QC testing, you batch release the TP. That is again 3-4 months. When everything is done, your package is ready, your regulatory is all set to find IND. The IND application, the IND journey at FDA is actually 2 months. You have to submit it 30 days in advance. FDA take 30 days to review it. Within a month and a half, 2 months, you get a safe to proceed if everything is fine. If there are more questions, there are meetings, there are correspondence which happen, and again, once and only once when FDA is fully they agree to your plan and the clinical trial as well, they will give a safe to proceed. But in terms of CMC activities, how to manufacture, how to achieve that drug product from day one on a very accelerated scale, it still takes 12 to 18 months. And to think that in the COVID pandemic, Novavax, sometimes they started working in January, and by this March, they actually have finished the phase 3 trial in UK is again a miracle. That entire exercise for any other vaccine or even for flu vaccine 2 years back would have taken 5 to 6 years. So probably, it was constant discussions they have with FDA, a lot of things they leveraged from their earlier work, that they were able to successfully achieve everything within 12 months. Some of the phase, like the phase 2, phase 3, you can always, FDA can always ask you to reduce it, do simultaneously a lot of trials. Again, pandemic calls for innovations at every level, and FDA was well aware and supportive of all the activities a lot of companies were able to, were doing for these vaccines. Now, when I said that analytics is very important, don't take my word for it. Sanofi, I'm taking a quote from Sanofi over here. Sanofi, as per Sanofi, they said 70% of the time of production of a vaccine is dedicated to quality control, which represents several hundred of tests. Again, these are vaccines, these are for healthy people, these are for kids, these are for newborn, parental, expecting mothers. So the quality is, utmost over there. The safety is there. And on the left-hand side, I'm showing the upstream development in just one bucket. And then, I'm showing the downstream buckets, harvest, and all the chromatography steps. And over here, as you could see, at every point, you have to send your sample for analytical testing. And again, the goal is to make sure that, specifications are being achieved, the quality is being checked, the intended purpose for each and every step is being achieved. And analytical testing is divided into in-process testing. It is divided into, on the left hand side, in the right hand side, I have 2 buckets. And the analytical toolbox, the top one is the batch release methods. Now these are the very critical methods. These are the methods which have specification. When a vaccine is released, it is released against these methods. And then in the bottom, I have the characterization methods, which are, because many of these molecules, recombinant protein-based vaccine, these are glycoproteins. There are a lot of glycans. Glycans are a very important part of testing. And a lot of the testing for glycans happen for the mass spec-based methods. for new new glycan analysis and epitope mapping. So all of these methods, the bottom half of this analytical toolbox is for information only. These are the characterization method. The top half is the release method, which are the must-have. These are very important methods which has to be passed within specification for FDA to give a go-ahead for a phase 1 trial or to commercial production. Again, everything start with identifying the product quality attributes. Once you design your methods, you ensure that methods are stability indicating. That is very important because you have to store your drug product throughout the entire timing of the clinical trial. And the drug product has to be stable, has to be efficacious, have to be pure. And you have the only way you can show is by doing the analytical testing, by ICH guidelines, throughout the clinical trial phase. And you learn more and more about analytical methods as you support process and formulation development. And then, you need to make sure that your assays are within, they are qualified enough, they're validated enough. They need to be validated to ensure that when you're testing them for the GMP material, they're good enough, that the results hold the trueness. Again, as I told you, a lot of aspect-based characterization is being done. They are a big part of the testing, but this is done for information only. New vaccines are coming up, which are using mass spec as a release assay. Amgen has done it, Biogen has done it, but again, it's a new and upcoming field. And finally, you have the method for identity, purity, concentration, potency, compendial, safety. Compendial is many assays. Once everything is done, you have a COA and submit it to FDA for testing. Over here, I'm listing the same. I'm listing the attribute and then the assays with the acceptance criteria. These are just for information only, but just to give you an flavor of it. How does a COA look like? And some of these tests are done for bulk drug substance. Some of these are done for drug product. Again, every assay is important. Every assay has to pass within the acceptance criteria. And the goal is to employ the appropriate analytical method, keeping the end goal in sight, which is you eventually have to release the drug product method. As I said in my earlier discussion, I discussed adjuvant. Now, adjuvant, I really give a lot of, already said a lot about adjuvant, but again, as I said, adjuvant is to support the protein-based vaccine. Adjuvant improve the immunogenicity. If there is no adjuvant, you actually have to give a very high dose of protein-based vaccine, which could eventually be, very toxic in nature, or you have to increase the number of boosts. Number of boosts is how many times you have to give the vaccine, like for Moderna or Pfizer, you are getting 2 shots. For a protein-based vaccine, even Novavax, there are 2 shots. If there is no adjuvant, you basically end up giving 5 or 6 shots to get that immune response, which is not allowed by FDA. So probably the adjuvant is a very key role in this protein-based vaccine. And some of the adjuvants which are already approved in US vaccines are alum, MF59, which is from Novartis, AS01, AS04 family, which is from GSK and CpG 1018, and now Matrix-M, which is Novavax is using. So over here, I'm not going to go much into this slide, but again, the bottom line is it's FDA 2,000 pages document is given to them, which has everything from development to all the testing, everything, how is it done, and probably, everything has to be done in the law, regulation, guidance document that has been outlined by them in order to receive a safe to proceed. The bottom line is that, when you are developing and manufacturing a molecule, in this case, a recombinant protein-based vaccine, you have to continuously in discussion with FDA. There are meetings, there are correspondence you can do, but then, it's an ongoing process. Over here, again, I won't go much into it. The goal is safety, and that is what FDA is looking for. Again, a lot of paperwork, a lot of review, and FDA really look for a lot of things in terms to make sure that the vaccine they are approving or they are giving go ahead, it's safe and safety and quality, because they are accountable to the participants in the clinical trial. Finally, on this slide, I'm just summing it up by, that there's a vaccine life cycle. It goes from research to phase 1, phase 2, phase 3 to commercial release. And everything actually, is done with the regulatory success in mind. That what is your regulatory success? What sort of molecule you are looking for? And all the designs for the process and the analytics are being done from the regulatory success in mind. Although, we do our best when we manufacture vaccines to make sure the vaccines are safe, but, only preclinical and clinical studies can truly validate the safety and efficacy of a product. And as I have, said many times in the presentation, safety is a priority during vaccine development approval. That is must have, because as I told you, safety is, vaccines are for healthy individuals. And vaccine is a cheap business, as I told you. They don't make much money because a lot of vaccines are being developed by public-private partnerships, and the government basically, buy all these vaccines. So not many vaccine companies survive actually. For me personally, I work at IAVI, International AIDS Vaccine Initiative. It's a not-for-profit. We are in our 25th year, and we work with a lot of product development. We are developing a lot of product in partnership with a lot of governments, other regional partnerships. We are based in New York. Our headquarters is over there. We have an antibody consortium at Scripps. We have a lot of professors over there who are actually, IAVI professors as well. We have an India lab. We have a human immunology lab in Oxford University, and then we have a lot of our own clinical research centers in East Africa. And in addition to HIV, we actually work on TB and many other diseases, as well. I don't need to impress you how important it is to get an HIV vaccine. It is said that even a very moderate efficacy, 50% efficacy vaccine will save almost 17 million lives, from now till 2030. For this webinar, I'm thankful to Dr. Antude, Eddie Saeed, Vadim, and Dr. Tom Hassel, and all the IAVI expedition team, who work together with me on these programs. And finally, IAVI is very grateful to acknowledge the generous support provided by the major donors, all around the world for all the work we do in the field of HIV vaccines and many other vaccines. And thank you. And if there are any questions.
Thank you, Vaneet. Please join me in thanking Dr. Sharma for his excellent presentation.
Yes, thank you.
All right, so we have a few questions that was sent through the chat. I will try to, compile them. If I miss your question, please do not hesitate to stop me and then ask your question. So the first question is, how is Pfizer-BioNTech and Moderna vaccine compared to the new Johnson & Johnson vaccine? This came from Rebecca Liu.
Rebecca, as I explained in my presentation, mRNA, Moderna, Pfizer are mRNA, synthetic mRNA in liposome. The Johnson & Johnson is a completely different vaccine. It is actually a double-strand DNA, which is in the genes format over, that virus is actually incorporated into a common cold virus. And again, you have to go back to the central dogma. It is the same central dogma. The end goal is same for both vaccines. Both of the vaccines wanted to make a spike protein in your body. Moderna or Pfizer, they do by using mRNA. And from mRNA, they just instruct the cells to make the same spike protein. And by using a viral vector, again, a different approach. Over there also, you have a double-strand DNA, basically, now making those mRNAs and instructing them to form the protein. So I would say that different chemistry, but end goal is same. And Moderna and Pfizer mRNAs are first in class, never been before. But the Janssen and Johnson & Johnson vaccine, they have Ebola vaccine was recently approved, and the similar vaccine was also approved by for Merck for Ebola. So history for those molecules. But again, both of them are very safe, very efficacious, and they differ in the basic chemistry, how they do it.
So the second question comes from Patty Spector. How long is a vaccine? I think what she's trying to say is how long does it take to get the emergency authorization?
Well, for COVID, we saw everything happen very fast because that was the need for that. But again, in a normal procedure, in a typical procedure, you don't get to EUA stage. EUA is only for emergency use. In a typical, for all the vaccines, they were approved, in a different format. For over here, you're on emergency and authorization, but they are, once you started immunizing people, there's a lot of data available. They are as good as approved. But again, the EUA is only for, the times like COVID pandemic.
So the next one comes from Bodhi Chaudhary. This is a little more technical. What types of equipment are used in cell and process development steps?
Well, again, I can tell you the different, sort of folks which come together, and that will help you understand the equipment. So in upstream, we have a lot of, process engineers. We have biotechnology folks who have those, cell lines and, all those things. So equipment which are there to do that, for the cell line development. Then in the downstream, you have your chemical engineers, your chemistry folks, your physics folks, your design engineers, and over there, much of it is chromatography. They use a lot of FPLC, the elder brother of HPLC, and then a lot of chromatography is done, viral inactivation is done by biotechnology. So again, it's a very complex procedure. But then there are very good review papers, for each and every unit which is published, which exactly tells what it needs to be done at upstream and downstream level. A very complex operation, a very costly equipment.
Now, Bodhi has a follow-up question, which is a slightly different area. If the drug substance that is mRNA-based is same in the Moderna and Pfizer vaccine, what differences are there in the formulation that warrant such a low freezing condition for storage for Pfizer, which is -80, compared to the Moderna vaccine?
Well, as I told you, the difference is on the liposome. Liposome technology is a very patented technology. And as I told you, there are going to be very future fights on those patents. And everything comes back to that liposome. Originally liposomes, everybody believed that liposome cannot be kept at low temperature. Liposomes have always to be kept at the room temperature. But, with adding those additional things, they were able to preserve it. The entire goal is to preserve it for longer times, But very recently, actually Pfizer now says that by doing some modifications in the formulation, they have been able to reduce it from -80 to once you get the vial out of -80, you can store it at a lower temperature. But everything is that liposome. If you will see in Moderna actually has SM102, which is an unknown, component. Only FDA and Moderna know what it is.
Excellent. Next question comes from Carol. Is there an animal model for COVID?
Again, mice model is the, humanized mice model is always used. But for COVID, I think for Novavax, they use a baboon, and they use, of course, they would have used mice model. But there is a Nature paper I referred in my presentation in that slide, which is from Novavax. I'm pretty sure they use baboons and mice. But again, humanized mice model is a model which can be used for recombinant protein vaccines, and for maybe for any other vaccine as well.
Patty has a second question. Can you elaborate on virus vector? What are those?
A vector is a device. Vector is a device. Vector means that there is a common cold virus. The virus which give us a common cold, which is a very sort of let's put it this way, harmless virus. So in a viral vector, what they do is they take the genetic information of the common cold virus, the virus which gives you common cold. And in that virus, they actually chop the virus and add the coronavirus in between. And the entire goal is that, because it is very important for our body to accept, for our cellular processes to accept the virus in such a way that they don't mount a huge immune response against the virus. So it has to be a very subtle response, done it very cleverly. So the way they do it is they basically confuse our body that this is a common cold, let it settle down. And in the middle of it, the coronavirus is able to do its work. And, it is able to produce a spike protein and generate the immune response. If you will be able to give and the coronavirus as such, then it has to be an inactivated virus, as is done by many other vaccines. So again, a different way, and the word they use is basically smuggling, that you are able to, that you're trying to smuggle the coronavirus in a common cold virus, confusing your body in order to accept it so that it can, give the desired spike protein for immune response to be generated.
Great. Next question comes from Sylvia. Will the Johnson & Johnson vaccine also protect people from the common cold?
Very, interesting question. I think yes and no, but more importantly, I think they don't want the protection from common cold. They want that, when the vaccine is given, you feel little sick, because that is the only way, when you feel little sick. A lot of people, when they go for the Moderna vaccine or when they go for other vaccines, they report, oh, I feel little sick. That is the good news. That means that the vaccine is doing its job. If you get a vaccine and you absolutely feel fine, then okay, maybe your body, is taking more time. But if there are, there is a response which happens, you feel a little sick, you're a little feverish. Probably the reason, if you remember, when we were young or young kids, when they get vaccine, they get fever, because that itself is an endpoint that the vaccine is doing its job. So I think the goal over there is not to get rid of common cold. For that we have other vaccines, but probably it's that can do. It's a mixed bag. You will be able to, interesting concept, but probably not the intended purpose for the vaccine, for this vaccine.
Perhaps we can take the presentation away so that we can see more people in here. We still have a few follow-up questions. We're keeping you here for at least another 5 minutes.
No, then I'm here. That's okay. So do you see what do you see now? Stop here?
I see the end of this. There we go.
Okay,
we are
good.
all
Thank
back.
you. Yeah,
We're all back. Okay. So the next comment is from Min. He's very appreciative. He doesn't typically see the manufacturing side of this type of research. He wanted to thank you.
Oh, thank you. Again, that was my goal of the presentation, because someone at my level, we don't do research. We don't design molecules. Those are done in universities by professors, by research. We only develop and manufacture. And I wanted to give that operational aspect of manufacturing, trying to answer the most important question where everybody's asking, where are the vaccines? Why they take that much time?
So 2 follow-up questions. One comes from Rebecca. What learnings from the COVID-19 vaccine can be applied to the research for AIDS?
Well, actually, it is the other way. Everybody is actually saying that all the hard work that was done for vaccines against, AIDS vaccine all these years is the reason why we have COVID vaccines. All the trial centers, all the special centers, testing centers, which NIH, universities, people, folks like, companies, they put together for HIV vaccines, they were used to make sure that the COVID vaccines are available that fast. Moderna vaccines, they are not able to come with a vaccine just like that. They have a history of working on Zika virus. They have a history of working on HIV vaccine. Moderna is actually working with us on HIV vaccine. So a lot of learnings from HIV has resulted in the COVID vaccine. And now the other, the opposite is also true, because with the approval of mRNA vaccines, now this is a new world where a lot of things are being done through mRNA. But again, there's always a lot of excitement when a new molecule is there. People tell me the same thing when the DNA came. There was so much excitement that DNA is going to solve all the issues. Similarly, mRNA is going to be there. Everybody wants to test mRNA for every pathogen. May not be same success as for COVID. COVID was a different type of virus, which is good for us. But then, it's a mixed bag. Folks learn from AIDS research, and now a lot of pressure and where they want to use mRNA, because mRNA has that inherent the way you can manufacture it that fast. That is one thing which everybody, people love fast things, like they love, so they want to make it fast enough to be tested in clinic.
Now, one other question. This comes from Min. How does the new COVID vaccine get approved for human trial without large animal studies? Is it usually the case from mice to human directly, or just go through chimpanzees in between for all drug trials?
So there are 2 aspects of this one, Min. If this would not have been a COVID world, this is the typical normal world of 2018, 2019. Then there are two aspects. If you are working on a novel platform, novel vaccine, over there the FDA asks you for mice data, of course, a humanized mice data, and then they also ask you NHP studies, in order to understand the safety, how much dose toxicity you have. And a lot of, in NHP, I think the monkey model is used for, and then there is a mice humanized vaccines. And COVID vaccines were different altogether. And I think, as I said, that at the end of the day, it was a decision which FDA took with a lot of these manufacturers. And a lot of these manufacturers were able to impress with FDA that, if we are able to do, if we end up doing everything we do in a typical normal world, probably it would take much more time to develop the vaccine. Now, what we can learn from, what are the leverages from the work which they have done in past? Moderna is not making an mRNA vaccine for first time. They've already done that for flu, not manufactured, but phase 1 study. They've already done a phase 1 for Zika. So they understand what is the, how does the vaccine behave in animals? How much in animal studies, most important thing is the dose. You have to do the dose ranging studies to understand the toxicity of the vaccine, how much doses you can give, how many times you can give. So if you can learn from the leverage of the past, as Novavax have done, Novavax must have gone to the FDA, and they would have, Novavax has a very rich history. They basically they're a 30-year-old company. This is the first time they're going to manufacture a product. They put all their energies in RSPF protein, which is for respiratory young kids vaccine. They invested everything. They basically got bankrupt in 2019. But all the learnings they have done from that work, which came very handy for this COVID work. So it is an amazing story, Novavax success story. Basically, the company was closing its door, and now we have the only company which basically have a protein-based product. So I think that this is a very interesting case study what FDA did over here for COVID vaccine, which is again good for everybody without cutting corners. Safety was still there, and trying to make sure that without cutting corners, the safe vaccines are given. Probably there will be a lot of papers published very soon about how and what FDA did, and all of these are very, very medical stories. How these vaccines were brought into manufacturing within a year or so.
Great. I think we have one last question, and that comes from one of our students. Her name is Rachel. Rachel is a student in her BA track, and she appreciates the information that she got from you. Her question is, do you see how this pandemic has affected long-term vaccine production in the future years?
Again, a very interesting question, because if you will, again, you being a bachelor, you're trying to understand where are the jobs for next year. The good thing is a lot of jobs in the vaccine industry, and the reason for that is that there are a lot of companies were already making a lot of vaccines. As I said, a lot of vaccines are given to the newborns, to the expecting mothers, for vaccine for everything. So there are, a lot of jobs which are already there, but now there's going to be a booming vaccine industry. And for that, the reason being, there are new forms of vaccine which have, come into the picture. These vaccines are going to be tested against each and every pathogen. These vaccines have opened the door for the cancer vaccines. So those companies, which are typically oncology companies, small molecule companies, now they are interested in mRNA-based vaccine. So in terms of production, in terms of, the more and more vaccines available for a lot of other pathogens, I think that is going to be reality, for individuals who are in chemistry courses and biotech courses. There are going to be a lot of jobs around. And as you would have seen in the presentation, like, being a little biased, analytical chemistry was, a typical, a lot of jobs for them, because there is, there's an analyst for every role. There is an analyst who tests the pH. There is an analyst who tests the appearance. There is an analyst whose only job is to do ELISA. There are 10 of them. There are analysts who do HPLC. So, these are the places, a lot of CMOs, companies are going to have a lot of jobs opportunities in the field of vaccine. The only goal is that, the right skill set, the right analytical instrument information. Even then, many of the entry-level jobs, are going to be for the freshers. You know, companies always give an opportunity for them to come and join and learn.
Excellent. Are there any other questions that you want to ask yourself? All right, well then, once again, thank you for joining in. Dr. Sharma, this is a really useful talk, and we appreciate your time with
thank
us.
You. Thank you, Saurabh.
Well, thank you very much. We'll call it a night. Again, if you have any questions, please send it to me. My email address is on the website. There are questions recording. We're going to try and make this recording available to public so that you can watch it later. Thank you.
Okay, thank you, Saurabh. Thank you for the opportunity. Have a good night.